When we usually picture a tumor, we tend to think of it as this kind of localized threat.
Right, just a mass of cells.
Exactly, like a mindless rogue mass of mutated cells just taking up space where it absolutely shouldn't be.
Yeah, causing damage just by sheer physical pressure.
Right. We visualize it as a static roadblock on a highway. But what if we've had that fundamental assumption completely wrong this whole time?
Well, that's exactly what we're looking at today.
Yeah, because what if a tumor isn't just a roadblock, but, a highly sophisticated rogue hacker, one that is actively tapping into the brain's own communication grid to literally fuel its growth?
It's a profound paradigm shift. I mean, honestly, it challenges pretty much everything we thought we knew about neuro-oncology.
It's wild to even think about.
It really is. The field is moving away from seeing certain cancers as just these isolated masses of sick tissue. We have to start looking at them as integrated parasitic networks, like an invasive secondary organ system almost.
Which brings us to the mission for today's deep dive. We are looking at a truly groundbreaking study published in Nature. It's titled, A Prognostic Human Brain Network for Diffuse Midline Glioma.
And the researchers pulled off a massive methodological feat here.
Oh, totally. They integrated data from hundreds of patients, mapped it against healthy brain connectomes, and then drilled all the way down to single-cell RNA sequencing.
Right, all to fundamentally rewrite our understanding of how these devastating childhood brain cancers actually operate.
Before we get into the weeds,
Yeah.
Can you just define what diffuse midline gliomas, or DMGs, actually are?
Sure. So DMGs, which actually includes the tumors historically referred to as DIPG, they are near universally lethal tumors in the central nervous system of children.
And they've been notoriously hard to understand, right?
Extremely. For decades, the clinical behavior of these tumors just baffled doctors. I mean, they would aggressively spread to distant, seemingly unconnected brain regions.
Like the frontal and temporal poles.
Exactly. And the mechanism behind that specific routing was just a total mystery.
Okay, let's unpack this, because we're going to take you on a journey today.
We really are. We have to look at the entire board to understand this.
Right. We're going to start at the macro level, looking at massive data sets of entire brain networks, and then eventually we'll zoom all the way down into the micro-level chemical wiretaps inside individual tumor cells.
And I think the best place to start is with the realization that the map we've been using for decades is, well, essentially useless for this disease.
The physical map, you mean?
Right, because in traditional oncology, there is this intense focus on geometry. Like, where exactly is the physical footprint of the tumor, and what is its total volume?
Right, the old real estate adage, location, location, location.
Exactly.
If a tumor is pressing on a critical structure in the brainstem, you'd assume the outcome is way worse than if it's sitting somewhere with a bit more, buffer room.
That's the logical assumption, and it's exactly what the researchers wanted to test rigorously.
So how did they do that?
They used a technique called voxel-based lesion symptom mapping, or VLSM.
VLSM.
Got it. So they gathered MRI scans from a discovery cohort of 125 children treated at Great Ormond Street Hospital.
Okay, so a really solid data set of kids with this specific cancer.
Yes. And the goal was to take the exact 3D coordinates of each patient's tumor and map it to see if precise physical location, or just total tumor volume, could predict whether a patient would have short-term or long-term survival.
So they're basically looking for the deadliest coordinates in the brain. What did the mapping actually show?
It showed absolutely nothing of prognostic value.
Right, really, nothing at all.
Nothing. The results were completely counterintuitive to traditional oncology.
Wow.
Neither the precise location of the mass within the brainstem or the thalamus, nor the sheer volumetric size of the tumor predicted how long the child would survive.
Wait, I have to pause here. Because in neurosurgery, millimeter-level location is usually everything.
Oh, absolutely.
Like, a tiny lesion in the wrong spot of the pons can be catastrophic compared to a massive lesion in, say, the frontal lobe. Why does that fundamental rule of physical real estate suddenly just break down with DMGs?
Because these specific tumors are not just occupying space. They are actively integrating into the local circuitry.
Okay, explain that. How do they integrate?
Well, preclinical animal models have revealed this highly sinister mechanism. These DMG cells are physically forming what are known as neuron-to-glioma synapses.
Wait, they are biologically wiring themselves into the healthy neurons?
Yes, literally. They are extending these tiny microtubes and plugging directly into the brain's electrical grid.
That is horrifying.
It is.
Yeah.
When the healthy neurons fire and release neurotransmitters, Those signals jump the synaptic cleft and depolarize the membrane of the glioma cells.
So the tumor cell is receiving the electrical signal just like a normal brain cell would.
Exactly. And that electrical shift, combined with paracrine signaling, where the tumor cells release and receive local chemical growth factors like BDNF and a protein called NLGM3, it acts as a direct molecular command for the tumor to divide.
To just rapidly proliferate.
Right. It's using the brain's normal activity to fuel its own growth.
Okay, so let me make sure I'm synthesizing this correctly before we move forward, because it's time for our first quick 5-minute recap for you guys listening.
Good idea. There's a lot of dense science here.
Okay, bullet points.
1.
DMGs are incredibly deadly childhood brain tumors. 2, against all logic, tumor size and precise physical location do not predict patient survival.
Right.
And 3, preclinical models show these tumors physically synapse with healthy neurons to grow. They plug into the grid.
Perfect summary.
It really makes you reconsider how we define cancer. I mean, have you ever thought of a cancer not as an isolated mass, but as a parasitic organ trying to join your nervous system? It's not just a broken machine part. It's a parasite actively feeding off the machine's normal operations.
Which perfectly sets up the next major hurdle the researchers had to clear.
Right.
If the physical location of the mass doesn't predict survival, they hypothesize that the tumor's connectivity to the rest of the brain might.
But mapping connectivity in a brain that is already distorted by a tumor must be incredibly difficult.
It's nearly impossible. Because the tumor has already warped the local architecture, you can't trace the normal pathways because the roads are already ripped up.
So how do you measure a network that's under active destruction?
You don't look at the sick brain. You look at a healthy blueprint.
Oh, that's clever.
The researchers developed a breakthrough technique called tumor network mapping. They took the exact coordinates of the patient's tumors and overlaid them onto a massive independent data set called the ABCD-1000.
The Adolescent Brain Cognitive Development Study.
Yes, exactly.
That's a database of resting state functional MRIs from 1,000 healthy 9-year-olds, right?
Correct. By projecting the patient's tumor locations onto this healthy pediatric connectivity map, they asked a really brilliant question.
Which was,
if we drop a pin exactly where the tumor is, what normal brain networks does that specific coordinate naturally plug into?
okay, to use a visual analogy for you listening, imagine the brain as a national highway system.
I like that.
Traditional oncology was just staring at the physical roadblock, like the tumor itself. Tumor network mapping takes the location of that roadblock and looks at the national blueprint to see where the traffic originating from that spot normally travels.
Right. They are mapping the downstream network.
And what did that map reveal?
The data revealed a very clear pattern. Tumors that were highly connected to a very specific distributed brain circuit were associated with significantly shorter patient survival times.
So the more plugged in the tumor was to this specific circuit, the more aggressive the disease.
Precisely.
And they named this circuit the DMG network. What actual neuroanatomy are we talking about here? Like, where do these highways lead?
Well, it originates in the pons and thalamus, but it extends far beyond the immediate tumor site. It strongly connects to the motor cortex, which handles voluntary movement, the insular and orbitofrontal cortices, which integrate sensory and cognitive processing, and the limbic system, involving structures like the amygdala and basal ganglia.
So if a tumor happens to germinate right on the on-ramp to this specific sensory and motor network, it has a fast track to aggressive growth.
Exactly. It exploits those specific pathways.
But, as we look at this study, I'm always kind of cautious about statistical flukes.
Of course, you have to be.
Finding a pattern in a highly specific cohort of 125 kids from one hospital is great. But how do we know this isn't just an artifact of that specific data set?
The researchers knew they had to prove this wasn't an anomaly, so they subjected the algorithm to rigorous external validation.
Meaning they tested it on other groups.
Yes, 2 completely independent groups of patients. One was a multi-center cohort of 125 patients from various global institutions.
Okay, good.
And another was a group of 38 patients specifically enrolled in the PNOC clinical trial.
And the DMG network predicted survival in both those groups too.
In both independent groups, yes. The degree of connectivity to the DMG network remained a robust, independent predictor of overall survival.
Wow.
And crucially, this held true even when they statistically controlled for confounding variables, things like the patient's age, sex, the extent of any surgical resection, and whether they had received chemotherapy.
So the network connectivity cut through all the noise.
It did. It was the strongest predictor.
I have another question about specificity, though. Let's say I have a completely different type of brain cancer.
Okay.
If it sits on this same DMG network, does it also grow aggressively? Like if we know the exact communication network the tumor is using, is this just a universally bad news network for any disease in the brain?
That was their exact next question. To test whether this was just a generic vulnerability in the brain, they took data from adult patients with glioblastoma or GBM.
Which is another highly aggressive, diffuse brain cancer.
Yes. They took the GBM tumor locations and mapped them to the exact same DMG network using the same methodology.
So let me guess, it didn't work.
Zero correlation. Literally zero.
That's fascinating.
The DMG network connectivity did not predict survival outcomes for adult glioblastoma patients whatsoever. This particular parasitic network is a uniquely DMG phenomenon.
Okay, taking a step back to look at the whole picture so far, we've basically thrown out the traditional focus on tumor size and physical location.
Yes, for this disease at least.
Instead, we have validated proof across multiple patient groups that these tumors survive and thrive based on their connectivity to a very specific, unique neural highway.
Exactly.
But this raises an immediate mechanical question for me. If the tumor is reliant on this network, does it just sit there and absorb the signals, or does it actually use the network to move?
That's the terrifying part.
Right. Like when we look at MRI scans over time, what is the actual geometry of how this cancer spreads?
Well, the clinical reality of how these tumors spread is exactly what makes them so devastating. To track this, the researchers looked at longitudinal data.
Scans taken over a period of time.
Right. They tracked 71 children who had progression MRIs taken within 2 months of their passing.
So we have the baseline scan and the final scan.
Yes.
When you subtract the original tumor map from that final progression MRI, what does the growth pattern look like? Is it isotropic-like, just expanding outward evenly like a balloon inflating in the brain?
No, it is highly anisotropic. It does not expand randomly. The tumors literally migrate and grow along the specific functional trajectories of the DMG network we just discussed.
Like a vine growing up a specific trellis.
That's a great way to picture it.
It's creeping along the exact neural highways it's wired into.
The imaging confirms that the functional network the researchers identified isn't just a statistical predictor of survival, it is the literal physical pathway the tumor exploits to invade the rest of the central nervous system.
That's just incredible.
And recognizing this pathway actually helped the researchers solve a long-standing clinical mystery, one regarding the age of the patients.
Right, the age of onset discrepancy. We know that pontine DMGs, tumors originating in the brainstem, they peak in incidence around age 5 to 6.
Correct.
But thalamic DMGs, which are the exact same disease but higher up in the brain, they peak later, around age 11 to 12.
Yes.
If it's the exact same type of cancer, why would the physical starting location dictate the age of the patient?
To solve this, the researchers had to look at normal, healthy neurometabolic development.
Okay, how do they do that?
They analyzed data from 18 FBG-PE scans of healthy children across different age groups.
For those familiar with neuroimaging, an FDG-PT scan is essentially measuring glucose uptake, right?
Yes, exactly.
It shows us where the brain is metabolizing the most sugar, which basically points to areas of intense energy consumption.
Right. In a developing child's brain, massive spikes in sugar uptake in a specific region signify a period of peak neurodevelopment.
So it's a proxy for intense synaptic pruning, myelination, network construction.
Exactly. It shows us where the brain's metabolic resources are heavily focused at any given age.
And when they cross-referenced those peak developmental windows with the tumor data, what did they find?
The alignment was honestly chilling. They found that the peak metabolic construction phase for the pons happens exactly at ages 5 to 6.
Wow.
And the peak metabolic construction phase for the thalamus happens exactly at ages 11 to 12.
The tumors are perfectly timing their emergence to exploit the specific brain regions that are undergoing maximum neurodevelopmental construction.
They are.
It's like a malicious contractor waiting until the exact moment the scaffolding goes up and all the expensive building materials are delivered to a site, and then just hijacking those resources to build their own structure.
That is precisely what is happening. The tumor requires the intense, hyperactive metabolic environment of a developing brain network to thrive.
So it's essentially a disease of development.
Yes. It is hijacking the normal, healthy, biological process of childhood brain development to fuel its own parasitic growth.
It is incredibly elegant biology, but just a devastating clinical reality.
Yeah.
Just knowing that a disease is essentially hijacking normal childhood development, change how you view its behavior? It definitely changes it for me.
Oh, absolutely. It changes everything.
All right, we're about 10 minutes in, so it's time for our second rapid-fire recap for you listening. Let's make sure we've got the foundation solid.
Go for it.
Okay, point 1. Tumor network mapping proves that brain connectivity predicts survival, not just tumor location.
Check.
Point 2. These tumors physically invade the brain by creeping along this specific DMG network.
Right.
And point 3. The tumor's age of onset perfectly mirrors the normal metabolic development peaks of those specific brain regions.
Yeah, exactly.
Okay, so we've established the macro view, we know the network, we know how it grows along the tracks, and we know it hijacks developmental metabolic spikes.
We do.
But here is where I want to push into the really deep science.
Okay, let's do it.
Because this highway theory is brilliant based on brain scans and connectivity models. But how do we actually prove that the tumor cells are listening to this specific network at a chemical level?
That's the million-dollar question.
Right. We can't just assume they're wired in without actually looking at the wiring.
You're entirely correct, and researchers didn't stop at the imaging. To prove this molecularly, they utilized a cutting-edge technique called single-nucleus RNA sequencing, or snRNA-seq.
Okay, doing RNA sequencing on brainstem tumor tissue sounds incredibly difficult, especially given how diffusely these tumors infiltrate healthy tissue.
It's very tricky.
So what does this snRNA-seq technique actually allow them to see?
Think of traditional bulk sequencing like listening to a massive choir. You get the average sound of the entire tumor mass, which blends the tumor cells, the immune cells, and the healthy neurons all together.
So it's just a lot of noise.
Right. But single-nucleus RNA sequencing allows us to isolate the genetic instructions, the transcriptomic profile active inside individual specific tumor cells.
Oh, wow. So it's like putting a highly sensitive microphone on a single singer in that choir.
Exactly. They use this to compare individual cells taken from high connectivity DMGs, so the aggressive ones heavily plugged into the network.
Right.
They compared those against cells from low connectivity DMGs.
So when you isolate the cells that are aggressively plugged into the network, what does their genetic signature actually look like? Are they building different proteins?
The transcriptomic profiles were significantly different. The high connectivity DMG cells were heavily enriched with what we call synaptic genes.
Meaning what exactly?
It means they were actively transcribing high levels of receptors for specific neurotransmitters.
Wait, so they aren't just physically sitting near the neurons, they are actively manufacturing the specific chemical receptors needed to catch the brain's messages.
Yes. They are building the receivers.
Which neurotransmitters are they eavesdropping on?
Specifically, they showed a massive upregulation of 5-HT2A receptors, which bind to serotonin.
Serotonin.
And M1 muscarinic receptors, which bind to acetylcholine.
Acetylcholine, got it.
They also showed a strong upregulation of gene pathways that respond directly to NLGN3, which is that paracrine protein released by active, healthy neurons we mentioned earlier.
So to bring back our hacker analogy, the tumor cells haven't just spliced into the physical communication lines. They have genetically programmed themselves to manufacture the exact chemical wiretaps needed to decode the serotonin and acetylcholine signals that naturally flow through that specific DMG network.
That's a perfect way to put it. It is a highly specialized cheme architecture designed to feed on normal brain function.
It is a remarkable level of adaptation. Just staggering.
It really is. And understanding that vulnerability leads us directly to the clinical implications.
Right, because if the tumor is fundamentally reliant on this network connectivity for its survival, what happens if we purposefully break that network?
And this brings us to a really fascinating and somewhat controversial surgical finding in the paper.
Controversial how?
Well, traditionally, DMGs, especially in the pons, are considered completely inoperable.
Right, because they are so diffusely spread through critical life-sustaining brain regions. You can't just cut them out without causing catastrophic damage.
Exactly, and that remains true for pontine tumors. However, the researchers looked at a retrospective cohort of 23 patients with thalamic DMGs who had undergone incidental subtotal surgical resections.
Let me stop you there. In neuro-oncology, a subtotal resection, meaning the surgeon goes in but has to leave a significant portion of the tumor behind because it's too risky to remove, is generally considered a failure, right? Or at least a suboptimal outcome.
Usually, yes. You want a gross total resection to remove all the cancer.
So why are we looking at cases where a tumor was deliberately left behind?
Because the data revealed something astonishing. For the patients with high connectivity tumors, that partial subtotal resection actually provided a significant survival advantage compared to patients who only had biopsies.
Wait, really? Even though there was still aggressive cancer left in the brain?
Yes.
How does that make sense biologically?
Because of what the surgery actually accomplished mechanically. By removing a chunk of the central tumor mass, the surgeons were inadvertently performing a disconnective surgery.
Oh, wow.
They were physically severing the tumor's connections to the broader DMG network.
They were literally cutting the tumor's phone lines.
Exactly.
By isolating the residual tumor cells from the serotonin and acetylcholine signals they rely on to divide, the surgeons effectively starved the remaining tumor of its electrical and chemical fuel, slowing the disease progression down.
Precisely. It serves as striking clinical proof that the network connection isn't just an incidental feature of the tumor. It is a critical vulnerability. The tumor needs the network to survive.
Bringing this all together, we've walked from the massive connectome maps of healthy children down to the serotonin receptors on single tumor cells, and finally to the surgical suite, where cutting those connections actually extends life.
It's a massive journey.
It really forces a complete reckoning with how we approach treating this disease moving forward.
It demands that we shift our entire therapeutic framework. We can no longer view these gliomas simply as rogue masses that need to be targeted with traditional, generalized cytotoxic chemotherapy or untargeted radiation. Right, the old blunt force tools. We have to treat them as integrated, parasitic networks that are actively exploiting the body's natural neurobiology and developmental timelines.
It changes the battlefield entirely. And, that brings me to a final provocative thought for you to take away from this deep dive.
I'm ready.
We've established that these aggressive tumors are heavily reliant on neurotransmitters, specifically serotonin and acetylcholine, to drive their proliferation.
Right.
If that's the fuel source, what if the future of cancer treatment for DMGs isn't just developing new, incredibly toxic chemotherapies? What if it involves repurposing existing, highly accessible neurological or psychiatric drugs?
It is honestly one of the most exciting implications of the study. We have decades of pharmacological research on neurotransmitter modulation.
Exactly. I'm talking about anti-serotonergic medications or anticholinergic drugs, medicines that are already sitting on pharmacy shelves, currently used for treating depression, anxiety, or movement disorders.
It's a completely different approach.
Could we use these existing drugs to chemically mute the tumor's ability to listen to the brain? If we can't always cut the network with a scalpel, how else could we block these chemical phone lines? Could the key to slowing down one of the most devastating childhood cancers already be sitting in a completely different aisle of the pharmacy, just waiting to be repurposed?
It's a profound question, and it's exactly where the research needs to go next.
When you finally realize the rogue hacker is relying on the city's own power grid to operate, you don't necessarily have to bomb the city. You just have to change the frequency of the grid. Thank you for joining us on this deep dive into the source material. Keep questioning the world around you, keep looking for the hidden networks, and we'll catch you next time.